Hey everyone, and welcome to our explainer on one of the immune system's absolute speed demons, the lectin complement pathway. We're going to break this whole thing down, step by step, and really zero in on the high-yield stuff you need for your exams. All right, let's jump right in.
So, here's a question for you. How does your body possibly fight off a brand new bug, a bacteria, a virus, whatever, that it's never ever seen before? I mean, you don't have any antibodies for it yet, right? So how in the world does it spot the danger and just attack? Well, the answer is all thanks to our innate immune system. And a huge part of that response is, you guessed it, the lectin pathway.
Okay, so what is the lectin pathway? Well, it's one of the three main ways our body kicks the complement system into gear. And if you need a quick refresher, the complement system is basically this army of proteins in our blood that complements or helps out our immune cells to destroy invaders. The lectin pathway is a critical piece of our innate first response team. It's the stuff we're born with, ready to go from day one.
So let's break down the key features here. First off, it's part of our innate immunity. That means it's always on, always ready to fight. It doesn't need to learn a thing. This is super important. It's antibody independent. It doesn't need to wait around for the adaptive immune system to make antibodies. It just goes. And here's a big tip for your exams. It's incredibly similar to the classical pathway. Keep that in mind because it's going to make remembering the steps way easier.
All right, first things first, in any battle, you've got to identify the enemy. So how does the lectin pathway actually spot a pathogen? It's all about pattern recognition, looking for a very specific signature on the surface of these microbes.
The hero of this first step is a protein called Mannose-Binding Lectin, or MBL for short. Think of MBL as the body's security guard. It patrols around looking for one specific thing, a type of sugar called mannose. Now here's the clever part. Mannose is all over the surfaces of bacteria, fungi, and viruses, but it's pretty rare on our own healthy cells. So if MBL sees mannose, it knows it's found an intruder. And big exam point here, structurally, MBL looks a whole lot like C1q from the classical pathway. That's not a coincidence.
So the initiation sequence is pretty straightforward. You can think of it in 3 quick steps. 1, MBL spots those mannose sugars on the microbe. 2, it latches on tight. And 3, this binding acts like a signal, calling in its backup, these enzymes called MBL-Associated Serine Proteases, or MASPs. Once the MASPs dock onto the MBL, you've got this active complex, and it's ready to unleash the cascade. And again, just like MBL is similar to C1q, these MASPs are the functional cousins of C1r and C1s from the classical pathway. See the path?
Okay, the bad guy has been identified. Now it's time to build the attack. This next phase is all about amplification, turning that one little signal into a massive, full-blown immune response through an incredible enzymatic chain reaction. It's like a tiny spark setting off a huge explosion. So that MBL MASP complex we just formed, it's now officially an active enzyme. Its first mission is to find 2 other complement proteins floating around, C4 and C2, and chop them up. This act of cleaving them is what really gets the ball rolling on the amplification process.
And this is where the magic really happens. The pieces that were just chopped off, specifically the C4b and C2a fragments, don't just float away. They stick together right there on the surface of the pathogen. And when they combine, they create a brand new, super important enzyme called C3 convertase. The name tells you exactly what it does. Its whole purpose in life is to convert or cleave C3.
Now this right here is the central amplification step of the whole complement system. That C3 convertase enzyme is an absolute workhorse. It's like a machine gun, just rapidly firing on C3 proteins, chopping them into C3a and C3b over and over again. We're talking about creating thousands upon thousands of these fragments, especially C3b, which is the key molecule that's going to drive the rest of the attack.
So what happens to all that C3b we just made? Well, some of it does something really clever. It actually sticks right back onto the C3 convertase that just created it. This addition acts like an upgrade, changing the enzyme's job. It transforms it into the next critical enzyme in the chain, C5 convertase. And its new target? You guessed it, the C5 protein.
Okay, by making that C5 convertase, the lectin pathway has done its unique job. Now, it merges onto a common freeway that's shared by all 3 complement pathways. This is the endgame. We're entering the final, destructive phase called the terminal pathway.
So, the C5 convertase does its job. It cleaves C5. The C5b fragment that's created is the foundation for the ultimate weapon of the complement system, the membrane attack complex, or MAC. This is so cool. C5b recruits C6, C7, C8, and then a whole bunch of C9 proteins. They all assemble together to literally build a drill that punches a hole right through the microbe's cell membrane. With this giant pore now stuck in it, water just floods in, and the pathogen swells up and explodes. Game over.
All right, this is super high yield for exams, so listen up. You absolutely have to know the difference between the lectin and the classical pathways. They're so similar it's easy to get them mixed up, so let's put them head to head and make this crystal clear.
When you boil it all down, the biggest difference between these two is the trigger and the speed. The classical pathway is part of that slower adaptive response. It needs antibodies, which take time to make. The lectin pathway? No waiting necessary. It's part of the immediate innate response. As soon as it sees those microbial sugar patterns, it's go time. This table lays it all out perfectly. Look at the top. The immunity type and the initiator are totally different. Classical needs that antigen-antibody complex, while lectin just needs MBL to find mannose. But then, look what happens next. The C3 convertase? Identical. The C5 convertase? Identical. It's like they take two different roads to get to the same city, and once they're there, they travel the exact same streets to get to the final destination.
Okay, let's wrap this up with your key takeaways. This is your exam cheat sheet right here. Number 1, it's antibody independent, part of your innate immune system. Number 2, the initiator is Mannose-Binding Lectin, MBL, which spots pathogen sugars. Number 3, it forms the exact same key enzymes as the classical pathway, C3 convertase and C5 convertase. And finally, number 4, it all ends by merging with the terminal pathway to build that microbe-destroying MAC. Got it?
So, if you like this video, then please like, comment, and subscribe.